So similarly to before, let's think about this same idea of adding all of these different things, and how would this change the carbon dioxide concentrations in a carbon dioxide analyzer. So in this one, we're looking at intact Chlamydomonas. So remember, if we're looking at intact Chlamydomonas, we can undergo both photosynthesis as well as cellular respiration. And we're going to start with just atmospheric gases being present in the chamber in the dark. So right here, we're starting in the dark. So without glucose being added, without any light available, our carbon dioxide concentrations are just going to stay the same. But now we're going to add light. So our carbon dioxide concentration is going to decrease because we're fixing that carbon using the Calvin cycle. But we don't have any additional glucose present quite yet. We're creating the glucose through the Calvin cycle, but now we're going to add exogenous glucose, extra glucose. So now if we have equal amounts, we could potentially be reaching our light compensation point almost. So we can kind of have one of two options. Either we're not going to change because our amount of photosynthesis is going to be equal to the amount of cellular respiration, or we can decrease a little bit more. Not quite at the same rate as we were with just light alone, because now we're undergoing both cellular respiration and photosynthesis. But our second option would be that we decrease just a little bit. So now we're going to add an uncoupler. So remember, even though we're talking about CO2 concentration, we add an uncoupler, we're rapidly undergoing our electron transport. So we're rapidly decreasing the amount of oxygen that we have. But if we're needing to go through the electron transport chain really, really quickly, we're also going to need to regenerate our FADH2 and our NADHs quite a bit. So we're likely going to be going through pyruvate oxidation and the Krebs cycle quite quickly. So in that case, we're going to increase our amount of CO2 in our CO2 analyzer. And now in the presence of excess light, remember this is when we're getting into that photooxidation segment, we're going to increase our amount of CO2 even more, because now we're damaging our photosynthetic electron transport chain units. So Hannah's asking if I can re-explain the uncoupler, 100%. So remember, uncoupler, we're rapidly going through electron transport. In order for us to rapidly go through electron transport, that means we're going to need to have a lot of FADH2 and NADH available. In order to have that available, we're going to have to rapidly go through pyruvate oxidation and the Krebs cycle, and that's where the source of our CO2 is. So if we add an uncoupler, we're going to increase our output of CO2 from those different cycles, because we're going through our electron transport chain very quickly. Asking if I could explain the glucose 100%. So blue is when our photosynthesis is equal to our rate of respiration. And that's why our CO2 levels don't change. But the purple, or the second line, this one right here, this would be if our rate of photosynthesis was greater than our rate of respiration. And so if our rate of photosynthesis is greater than our rate of respiration, then our CO2 concentrations are going to decrease still. All right, let's practice this. So again, 1 minute to do this one. Yeah, so Megan's asking if I can re-explain gross photosynthesis. So I'm going to do that before we get into this question. Awesome. So when we're thinking about gross photosynthesis, this is literally just asking how much oxygen am I creating and how much CO2 am I fixing. But if we're thinking like an organism like Chlamydomonas or like a plant, we have both chloroplasts and mitochondria. So it's not enough to just think about how much photosynthesis is happening, like we can when we're thinking about, just like a human cell, because we only have mitochondria there. Now it's a little bit more complicated because now we need to think about how much photosynthesis is happening, as well as how much respiration is happening. And that's when we're thinking about our net photosynthesis. So if we're thinking in terms of carbon dioxide concentration, we're going to be decreasing our carbon dioxide in our surroundings with photosynthesis, but we're going to be also simultaneously increasing them through respiration. So we need to take into account both of these things. All right, so I don't know if that changed anybody's mind in terms of what they wanted to answer for this one, but let me know what you're thinking. Couple more seconds. All right, so we're all kind of along the same page. Most of us are in agreement that 1 for sure is correct, but we're not entirely sure if 2 and 3 are correct. So we want to know which of the following statements are true for intact Chlamydomonas being measured in a carbon dioxide analyzer. So saying in the presence of light and carbon dioxide, gross photosynthesis is greater than the rate of respiration. That is true. Awesome. Everyone got that one. Because we don't have excess glucose yet. So our rate of just carbon fixation and oxygen production is going to be more than the rate of carbon dioxide being given off. Cool. Number 2, carbon dioxide levels in the analyzer increase in the presence of excess oxygen. So when we're thinking about excess oxygen, this would mean that we're undergoing more respiration. So we have more respiration happening. That means we're going to have likely more NADH and FADH2 being required. And if we have more NADH2 and FADH2 being required, we're going to have to undergo those Krebs cycles and oxidation of pyruvate more, and thus would increase the amount of carbon dioxide. So 2 is also correct. Number 3, in the presence of light, carbon dioxide, and acetate, gross photosynthesis is less than the rate of respiration. So we're thinking about light and carbon dioxide, that's going to increase our rate of carbon fixation. But if we're in the presence of acetate, again, because we're skipping those steps of glycolysis and pyruvate oxidation, we're likely going to be increasing our carbon or CO2 levels in the surroundings because we're going through the Krebs cycle really, really quickly. So 3 is also correct. Number 4 states that carbon dioxide levels in the analyzer decrease in the presence of excess light. Well, likely they would do the opposite. They would likely increase. So 4 is incorrect, leaving us with answer choice B. Yeah, so Terry is asking if I can explain number 2 again. So if we're thinking about what, I want us to really think about how would those ratios change. So in the presence of excess oxygen, that means we have a lot of available electron acceptors at the end of mitochondrial electron transport. So if we have a lot of these electron acceptors, that means we're going to be able to go through electron transport more quickly. If we're going through electron transport more quickly, we're going to need more NADH and FADH2s. And if we need more of those molecules, then we're going to need to go through the Krebs cycle more quickly. But the Krebs cycle is one of our sources of carbon dioxide. So if we're going through the Krebs cycle quickly, then we're going to increase the amount of carbon dioxide in the analyzer.